Okay, so it's very early morning, 5 a.m. I wake up like 1 hour back, but still feeling sleepy. All right, so no problem. So here's the thing that welcome to the week one biochemistry, and let's see what we got here. So before jumping into the topics, I want to discuss the different aspects of questions you get in your biochemistry. Uh, the biochemistry, what I think, is the basic for everything. Like every topic, you go into the physiology, you go into the pharmacology, you go into the specifically the pathology. A lot of things will come from the biochemistry. So it's better to start with the biochemistry. And that's why realizing this, the first aid book, usually my first aid book, the 2016 version, it had the biostats in the very beginning. But the later versions of the first aid books, they have the biochemistry in the beginning. And this topic, this biochemistry, can ask you questions usually from the molecular biochemistry, like the DNAs, the replication, transcription, translation, this kind of thing, and very minute details of these things. And also they ask you questions from the genetics, a lot of questions from genetics, like the hereditary charts, the family trees, not the family trees actually, the hereditary charts, and the inheritance of disorders in autosomal or X-linked or mitochondrial way. And they ask you some questions from the genetic terms, like there is different genetic terms that we need to discuss. So we'll go one by one, and also very importantly, they ask questions from the metabolism section, very important, and vitamins and nutritions, so many. And usually you get 5 to 10 questions in your step one exam that are directly from the biochemistry. At least 5 questions you get in your main exam. So it's very important. Now, this lecture that I prepared is from the first aid, from my notes, my own notes from UWorld, and also some supplementation from Pathoma. They are all together compiled, and they are arranged as is in your first aid 2018. So when you read them along with your first aid, you'll see that these topics will come as is, as the sequence as in your first aid. So it will be helpful for you when you read your first aid along with the notes. And I hope you already got the notes. If you don't, I'll send you the notes after the class, because I think if you joined later, I didn't send you the notes yet. But this handout, I'll send you after the class, this note. All right, so first is the molecular biochemistry. Your first aid book starts with the description of the structure of the DNA, and DNAs are very sophisticatedly structured in our cell, and they are inside the nucleus, as you know, but there's some other DNA called mitochondrial DNA. We are not going into that. Then usual DNA that is inside our nucleus is formed into something called a chromatin structure. And how they are structured here, as you can see, that uh, they are structured as a supercoiled fashion. This ball-like structure, these balls or beads, whatever you say, these orange balls, these are formed by the histone proteins, and this green structure is our DNA, which are coiled twice, and again they are clustered together to form this chromatin structure. Now, as you can see, there is something called heterochromatin, and there's some euchromatin. The heterochromatin is what? The chromatin structure where they're super jam-packed. They are so much tightly packed that is called a heterochromatin. And euchromatin, as you can see in this picture, they're loose. And also here you can see the acetyl group. Some acetyl group is pulling this euchromatin apart. And one idea you can get from here, that if you acetyl this, or you do the acetylation of these histone groups, there will be loosening of this structure. So you might ask me, what is different? What is wrong with if the DNAs are clustered, or what is wrong if the DNAs are loosely packed? All right, so this is important because if you want to run some procedures on the DNA, like the DNA, they convert, or they get copied into another DNA. We call it replication. And also, this DNA convert into RNA. We call them transcription. To run that procedures, you need to loosen this DNAs up. You need to loosen this DNAs up so that the enzymes can sit in this space and do their job, do the replication, transcription, and this kind of thing. So this loosening up is important. The heterochromatin on the other side, you can see they are so tightly packed that the enzyme has no chance. If some enzymes comes here, they cannot sit here. They try to sit here, they cannot sit here. So this is important to loosen the DNA up to do these procedures. Now here's some information, as you can see, that remember, our DNA is negatively charged, because DNA, I will show you a structure of DNA, that it got phosphate, and phosphates are negatives. So DNAs are overall negatively charged. But these histone proteins, these are positively charged, and this histone, they are formed by amino acid, mostly lysine and arginine. Very important, because this is sometimes confusing. If the question asks you, okay, what is a more prominent or more abundant amino acid in histone? It's very intriguing that they give you the option of histidine. What I had problem with, they usually ask that what is the most prominent amino acid in the histone. They give options like lysine, arginine, histidine, and if you see histidine and you think that, okay, histidine somehow sounds like histone, so let's pick histidine. But actually histones are not rich, or they don't contain the histidine. They're rich in lysine and arginine. These are positively charged amino acid. Now, what type of histone you have in this chromatin structure, that's important. In these bits, there are 4 types of histones you have. What are those histones? H2A, H2B, H3, and H4. These 4 types of histones are clustered here in these bits. And remember, each of them has 2 copies. That means 2 copies of H2A, 2 copies of H2B, and so on. So there are 8 copies of histone in these bits, and that's why they also call histone octamer. Octamer means 8. Octa means 8. There's another histone that is called a linker histone, and that resides here in between the strings, and they stabilize these loops. These loops of coils of DNAs. They don't form the actual bead, but they remain in between these beads. That histone is called H1 histone, and that has a name called linker histone. Now, our DNA loops twice around this histone octamer to form the nucleosome. This whole structure, the histone bead, and the DNA around it together form the nucleosome. And this nucleosome, uh, joins together, and they form this histone. Nucleosomes joins together and form this structure of chromatin. Now, this chromatin also contains non-histone proteins such as enzymes that are required for DNA replication and transcription. That is very important. So in chromatin, if someone asks that chromatin, what type of protein this chromatin got? This chromatin, they have histone proteins, you already know. They have also non-histone proteins, such as those are required for DNA replication and transcription. So these proteins, these enzymes also sit there in the chromatin structure, and they happen to come to work when they are necessary. All right, so in the next page, you see that some information about DNA methylation, histone methylation. I hope you already know it, that if you add some methyl group to the DNA, if you add some methyl group to the DNA, you can see they can cause imprinting. When we go into discussion of the genetics, you'll find more about imprinting. Imprinting is what? To silence something, to imprint something. So if you need to imprint something, or to silent, or to inactivate, simply inactivate something, you need to put the methyl group on that DNA. And one thing is important: if you think that the female body chromosomes, female cell chromosome, there are two copies of X chromosomes. But if you know the function of the X chromosomes, they are mostly involved in enzymes production, or coding for enzymes, this X chromosome, because when you see the disease, X-linked recessive disease, mostly related to the enzymes of the body. We usually need one X chromosome, male and female, just one X chromosome. So why the females have two X chromosomes? This is the nature, and also they don't need two copies of X chromosome. So what happens that in the female body, one X chromosome gets inactivated. One X chromosome gets inactivated, and this thing is called the formation of the bar bodies. The X chromosome that is inactivated in the female is called the bar bodies, BARR, bar bodies. And that thing also is done by methylation of the DNA. And also here you can see the repression of the transposable elements. What is transposable elements? This is a satellite of DNA. What is satellite of DNA? Suppose you have a whole gene. You have a whole gene and, whole copy of DNA, and there is a small sequence of DNAs. Those DNAs can change position. Those DNAs can change position. When we go into further detail, you'll see that in our genes, in our DNA sequence, there are some elements. Those are called enhancer, those are called promoter, those are called silencer. This can change their sequence, this can change their position, they can transpose. That means they can go place to place and do different function. They are necessary for the normal physiologic function, and at the same time, if they are mutated, or they are not working properly, they can cause carcinogenesis or different mutation. And these transposable elements are also repressed by this DNA methylation, which mutes the DNA, that means which silence the DNA. And also aging and carcinogenesis. In the modern era, it is thought that aging is a disease. It's not a natural procedure. I don't believe that. I think aging is good, but whatever, the science thinks that aging is a disease, and this aging is also, scientists think that methylation plays a role in this procedure. And also carcinogenesis, that means cancer formation. Now, histone methylation and acetylation, that is another story. This is not entirely like the DNA, because as you can see, the methylation of the histone, they can cause repression or activation. So they can either cause repression, they can cause activation. And acetylation, if you do acetylation of the histone, that relaxes the DNA coil. And remember from our picture of the very first slide that this acetyl group is actually relaxing these coils. All right, so give me 2 minutes. I have a call from a family member. I'm not sure what happened in this morning. Just give me 2 minutes so that I can check what is happening. Sorry for that. All right, so my grandmother is suddenly got sick and she is vomiting, so I might need to stop early. But let's proceed for some time. Then we'll stop for today's class. All right, so here you can see that the nucleoside, those who attended late, okay, uh, no one attended later. So, all right, so here you can see the two structure that is the very basic of the DNA. And here you can see one is called the nucleoside, one is the nucleotide. So what is nucleoside? Nucleoside is formed by the nitrogenous base plus sugar. The nitrogenous base is the uh, base we usually see that the purines and the pyrimidines. I hope you already know that there are some purines and there are some pyrimidines, and the purines are usually adenine and guanine, and the pyrimidines, those are necessary for DNA and RNA synthesis, is cytosine, thymine, and uracil. You should remember in RNA there is uracil and in DNA there is thymine. There are some exception, but usually the DNA contains the thymine, the RNA contains the uracil. And also I should remind you that this thymine is sometimes confused with thiamine. This thiamine is vitamin B1, but this thiamine is purely the pyrimidines, right? So now, as you can see that when this nitrogenous base adds up with some sugar moiety, this sugar moiety can be a ribose sugar or it can be a deoxyribose sugar, depends on which part they are going. Whether they are going to make DNA, they need deoxyribose sugar. If they need to make RNA, they are ribose sugar. These are nucleoside, and if you add some phosphate with them, they becomes nucleotides. Nucleotides. So adenine, suppose if I give you an example, adenine is a pure nitrogenous base, and it's a purine. And when you add a sugar with it, it becomes adenosine. So adenine within sugar, it becomes adenosine. That means it became a nucleoside. When you add a phosphate with it, it becomes adenosine monophosphate. So this nitrogenous base, let's take into account, this is adenine. You add the sugar, it becomes adenosine, right? And when you add the phosphate, it becomes adenosine monophosphate. 2-phosphate, adenosine diphosphate. 3-phosphate, adenosine triphosphate. So that's how this thing works. That first there is the nucleoside, then it becomes nucleotide. Now, one thing is important before going to the structures. As you can see here, say DNA structure. If I zoom out a little bit, you can see the double-stranded DNA. Uh, uh, we don't need to learn the structures in proper. The structure I showed you earlier of the purines and pyrimidines, you don't need to remember them. But there's something like in your first aid, you'll find that, okay, this, if you deaminate it, you make this. If you deaminate that, if you methylate this, and you make this. Sometimes those are necessary. Go over them and have a look at how this thing, but those are pure memorization. Now here you see that the double-stranded DNA, and here is the backbone of the DNA. Here's the sugar. Here's the deoxyribose sugar, and here's the phosphate group, as you can see. And here you see these bases, this nitrogenous bases, and they are forming bonds. And one rule is A binds with T. That means adenine binds with thymine, and cytosine binds with guanine. So this is a rule. This is a rule of thumb, you can say. After following this thing, if I go on the top, you can say that the bonds, which bonds are more stronger and which bonds are not that stronger. The GC bond, the guanine and cytosine bonds, they have 3 hydrogen bonds. And they are very strong, and you can understand why. 3 hydrogen bonds are much more stronger than 2 hydrogen bonds, and AT bonds, they got 2 hydrogen bonds. So GC, 3 hydrogen bonds, and AT, 2 hydrogen bonds. All right, and now there is the purine synthesis, that how these purines are getting synthesized. In your first aid, you'll see this picture, that de novo purine and de novo pyrimidine synthesis, and this is a gem. You need to go over and work on this thing, so you won't miss 1 or 2 questions from out of this. But as you can see, that how they are formed, this purine and pyrimidine, they are formed with the help of this sugar. They requires some amino acid, like as you can see on the right hand side, the purine base production are reused from salvage pathway. That means the purines can be produced de novo. De novo means what? Newly synthesized. And from salvage pathway, that means they are already produced and they are recycled, and they form some purine base from those recycled products. So now here you can see that this ribose 5-phosphate is necessary, and with the help of this enzyme, sometimes enzyme names are necessary, like here this enzyme name PRPP synthetase. PRPP, the full form of PRPP is phosphoribosyl pyrophosphate. This enzyme converts this ribose 5-phosphate to PRPP. This is the phosphoribosyl pyrophosphate. And on the right-hand side, you can see that following some steps, those are not shown here, but I'm going to discuss it. They are converting into IMP, something called IMP, also called inosine monophosphate. And this IMP, with the help of some other enzymes, they converts into AMP and GMP. That means your purine nucleotide. AMP is what? Adenosine monophosphate, and GMP is guanosine monophosphate. So that's how you get the purine. Very simple, but usually they ask question from purine salvage more than the purine de novo synthesis. But as your first aid guide and as your this lecture guide, let's discuss this purine synthesis a little bit. As you can see, this ribose 5-phosphate from the pentose shunt, we're going to discuss this pentose shunt later. This ribose 5-phosphate we got, and this ribose 5-phosphate, this sugar or sugar moiety, as you can see, it got a sugar and a phosphate group. With the help of this enzyme, PRPP synthetase, this is converting into PRPP. And they need some other enzyme to convert into IMP, which is not shown in your book, but is shown here, that you can see that they got something called glutamine PRPP amidotransferase. This enzyme is a midway enzyme. The PRPP converts into 5-ribosylamine with the help of this enzyme, glutamine PRPP amidotransferase. And they convert into 5-phosphoribosylamine. And there are some other enzymes that is working here, not necessary for us for step 1 examination, that converts into IMP, the inosine monophosphate. And then from the IMP, as you can see, that they can convert into both ATP, or sorry, first AMP and then GMP. And from there, they can convert into ATP and GTP. On the left side, you see that they convert into adenyl succinate with the help of this enzyme, adenyl succinate synthetase, not necessarily to remember. And from this adenyl succinate lyase, this enzyme convert them into AMP. On the other hand, and the IMP converts into, through the enzyme IMP dehydrogenase to XMP, that means xanthine monophosphate, and with the GMP synthase, they convert into GMP, but not necessarily you need to remember this. If you remember this enzyme name PRPP synthetase, better if you remember this enzyme name PRPP amidotransferase and IMP dehydrogenase, that's all. These 3 enzymes will test you, sometimes will test you on your questions. So that's how you get the de novo, that means newly formed purine. All right, so next thing is de novo pyrimidine synthesis, that how the pyrimidine works. The pyrimidine are processed. As you can see, that pyrimidine base production, they requires aspartate. All right. Now, here you see that they requires glutamine, they requires carbon dioxide together with the help of ATP, with the help of this enzyme carbamoyl phosphate synthetase 2. Remember, this is carbamoyl phosphate synthetase 2. And there is another carbamoyl phosphate synthetase 1, and that we'll discuss in urea cycle. So when I go to urea cycle, I'll ask you that what was the enzyme that we discussed in the de novo pyrimidine synthesis? So in case of de novo pyrimidine synthesis, it is carbamoyl phosphate synthetase 2. But in case of urea cycle, that is carbamoyl phosphate synthetase 1. Now this glutamine with the carbon dioxide converts into carbamoyl phosphate, and this carbamoyl phosphate converts into orotic acid with the help of aspartate. But here is also some steps. Those are not shown in your picture, and that is shown in this picture. As you can see, in the left-hand side of the picture, the pyrimidine synthesis. Here is the carbon dioxide, glutamine, and ATP, and carbamoyl phosphate is formed here. Then with the aspartate, they form the carbamoyl aspartic acid. They form the dihydroorotic acid. Then there is orotic acid, and then there is this thing. From orotic acid, there is UMP. So these steps, this dihydroorotic acid, carbamoyl aspartic acid, is not shown in your book. But if you want to know it, if you want to know it detailed, you can look at this picture. Now, here's another thing that you can see, that this drug is inhibiting some steps. This leflunomide is inhibiting some steps in this reaction to convert into orotic acid. So these drugs, when we discuss the drugs in respective sections, you'll see that how they work and where they are needed. Now, this orotic acid, with the help of our PRPP, look, this PRPP is needed in both sides. It is needed in the purine synthesis part. It is needed in the pyrimidine synthesis part. This PRPP and orotic acid converts into UMP, that is uridine monophosphate. And here you can see that uridine is already here. Uridine is one of the pyrimidine. You already got it. And if you can see that there is something called impaired in orotic aciduria. So what is orotic aciduria? Orotic aciduria means if you block some steps here, some enzyme is stopped here, there will be accumulation of the orotic acid, because this orotic acid cannot convert into UMP. So there will be accumulation of orotic acid, and there will be a lot of orotic acid in the body, and that orotic acid will go out through the urine, and that condition is called orotic aciduria. So we can get orotic aciduria by this way or other way. If there is increased production of orotic acid due to any other condition, we're going to discuss it in the urea cycle. We can get orotic aciduria on that case also. But here, remember that if you block something here, impairment some enzyme in this pathway from the orotic acid to UMP, you get more orotic acid, and that orotic acid goes out through the urine, and that causes orotic aciduria. Now this UMP converts into UDP, or uridine diphosphate. But before going there, look at this thing, that orotic acid is converting into UMP. And if I tell you the specific enzyme that work here is called a UMP synthase. This UMP synthase converts the orotic acid to the UMP. So if someone has defect in UMP synthase, they can have orotic aciduria. And it can be a hereditary condition. That means it can pass from generation to generation through inheritance. When some disease passes from generation to generation through inheritance, that is called hereditary condition. And some words about this condition, like as you can see, it's an autosomal recessive fashion. That means both the mother and the father need to have the disease to pass it on to the children. And there is problem in de novo pyrimidine synthesis, as we already saw. And what is the symptoms of this condition? There can be physical and mental retardation. There can be megaloblastic anemia. I'm gonna say why there is megaloblastic anemia, and there is increased urinary uric acid level. Now, how you treat this condition? You treat this condition with uridine supplementation. You give the patient uridine, and when you give the patient uridine, uh, there is another mechanism in the body through which the uridine can be converted into UMP, because you need the UMP, right? As you can see in this picture, you need UMP, not the uridine. You need the UMP. There's some enzyme in the body that is called kinase enzyme. When we go discuss the metabolism, you'll see that the kinase enzymes, whatever kinase is there, the job of the kinase enzyme is to add phosphate to something. So here is some UMP kinase or nucleoside kinase is there that converts the uridine, the given uridine, into UMP, and that UMP can be used to proceed with this cycle. Now, as you can see, this UMP converts into UDP, just adding another phosphate, and this UDP is going into places. Like this UDP can be converted into deoxy UDP that can be used for DNA synthesis, but we don't need uridine for the DNA. We need TMP, that means thymine or thymidine for DNA. So what happens is DUDP converts into DUMP again, and then with this very important enzyme, thymidylate synthase, it converts into DTMP or deoxythymidine monophosphate. And also you can see that the UDP can go another place and it can form the cytidine. The enzyme we don't need to remember, that we don't need to know what is the enzyme working here. So this is a crucial step. From UMP, you got UDP. From UDP, you got CTP. From UDP, you got DUDP. From DUDP, you got DUMP. And from the DUMP, you got DTMP. And this enzyme is very important, thymidylate synthase. Another important enzyme is hydroxyurea, that is ribonucleotide reductase. Ribonucleotide reductase. And this enzyme, by stopping this ribonucleotide reductase, they are actually stopping the whole procedure. This whole procedure will be stopped. If you use the hydroxyurea, this whole procedure will stop, right? And by stopping this, they're actually preventing both purine and pyrimidine synthesis. And you might ask me how it can stop the, okay, you can understand that the purine or the pyrimidine synthesis is stopped by hydroxyurea, but how the purine synthesis is also stopped by just preventing ribonucleotide reductase. How this can happen? Because if you look at your book, The First Aid, when you read it, not necessarily now, that this hydroxyurea, by inhibiting this enzyme, they're inhibiting the synthesis of these products, right? And one of the important products is this tetrahydrofolate. And this folate group, or this folate group, methylene tetrahydrofolate group, they are necessary for carbon donation. When you go into further details of biochemistry, you see that these folate groups are necessary for carbon donation. That means they donate carbon to some molecules. So when this hydroxyurea stops this enzyme, they're stopping this folate group also. And when the folate group is stopped, they cannot donate carbon. And the purines, if you look at the structure from the very, purines, if you look from the structure from the very first page, you'll see that they are carbon-rich compounds, right? Here is carbon, here is carbon, 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 carbon. Some of the carbon needs to be donated from the tetrahydrofolate, or some of the carbon needs to be transported through the tetrahydrofolate. So by stopping the enzyme ribonucleotide reductase, you are not only stopping the pyrimidine synthesis, you are also stopping the purine synthesis. So that's why this is necessary. This is important to understand that by giving hydroxyurea, you are inhibiting both purine and pyrimidine synthesis. Now here you can see this thymidylate synthase converts DUMP into DTMP. At the same time, this cycle is running. There is conversion of N5, N10 methylene tetrahydrofolate to dihydrofolate, and this dihydrofolate, with the help of this another important enzyme, dihydrofolate reductase, to tetrahydrofolate. And this tetrahydrofolate again converts into N5, N10 methylene tetrahydrofolate, and this cycle continues. Now, if you block this one also, this thymidylate synthase by 5-fluorouracil, if you block this thing, 5-fluorouracil, this whole conversion will not happen. This whole conversion will not happen unless someone is supplied with folate. If you use this drug, this 5-fluorouracil, it's an anti-cancer drug. It can block the folate synthesis also. At the same time, it is blocking the thymidylate synthesis. It blocks the folate procedure. So unless someone is supplied with folic acid. And also you can see that these drugs, methotrexate, anti-cancer drug, or the drugs used in arthritis, the TMP, that means trimethoprim, that antibiotic, or pyrimethamine, the antiprotozoal agent. These drugs can also inhibit the dihydrofolate reductase, and if they block this step, you can see that this whole step is not running, and if this step is not running, this TMP synthesis is not occurring. So what is the point of using these drugs? Or what is the point, as I told you earlier, that there is megaloblastic anemia in aortic aciduria? How this can happen? All right, if you block here, suppose take into account the aortic aciduria. If you block here, the UMP is not there, the UTMP is not there, the CTP is not there, and these are necessary molecule for DNA synthesis, right? So think about a cell that cannot generate or that cannot replicate its DNA. It may have one DNA, but to make another copy of the DNA, you need these molecules. You need these materials, prerequisite to convert it into another copy of DNA. If some cell does not have these molecules or does not have this prerequisite, they cannot replicate. And to divide a cell into two, suppose a cell is here and it divides into two, it needs to divide its genetic material. At the same time, it needs to divide its cytoplasm. So when a cell cannot divide its genetic material, if it cannot divide its genetic material, what will happen? This cell cannot divide and this cell becomes big. That means it becomes a mega cell. And if it happens in the blast cells of the RBC, that means early phase of RBC, those cells becomes big and we got megaloblastic anemia. And you already know that in megaloblastic anemia, we don't have only RBC, big size RBC. We also have problem with the neutrophils. We also have problems with the megakaryocytes because those cells becomes big also. So by stopping this any step, either in aortic aciduria or using these drugs, there's a chance of having megaloblastic anemia. All right, so now next proceed into here, as you can see, some words about methotrexate. Methotrexate is a folate antagonist, as we saw earlier, that it is used in this place by inhibiting dihydrofolate reductase. Methotrexate is used here. And this methotrexate is a folate antagonist, is a drug of choice for non-surgical treatment of early unruptured ectopic pregnancy. I put an end here. This end should not be here. All right, so can I remove it? Sorry. Not sure how to remove it. All right. Whenever you read it, this is a mistake. All right. So methotrexate, the folate antagonist, is the drug of choice for non-surgical treatment of early unruptured ectopic pregnancy. So it might happen that you need the folate for the growth of the fetus, right? So when you block the folate, that fetus cannot grow. And if someone has unruptured ectopic pregnancy, suppose a woman have, usually the vignette is like this, some women have a history of PID or pelvic inflammatory disease, was not treated well, and now comes to you with the urine beta HCG positive, that means it's a pregnancy, and you do the ultrasound, you find the pregnancy is in an ectopic place, not in the normal uterine pregnancy. It may be in the fallopian tube. And that pregnancy can have a chance to rupture. If it is not ruptured, you can prevent the rupture by giving this patient this methotrexate. But ultimately, you need to surgically remove that pregnancy. All right, now let's proceed to the purine salvage pathway. That means how you recycle the purine. We discussed how you synthesize purine newly, but now here is how you recycle the purine. So here you see in the purine salvage that there is first there is the synthesis of purine. As you can see, a little bit of synthesis, the ribose 5-phosphate with the PRPP synthetase enzyme. It is converting into PRPP, and from PRPP you got the IMP, the inosine monophosphate, and from the IMP you got the AMP, you got the GMP, right? And from there the nucleic acids are formed. And as you can see, the arrows, they are showing that the nucleic acids are formed with the GMP, but when the nucleic acids are metabolized, they convert into these materials also, AMP and GMP. Now this IMP converts into inosine. This IMP converts into inosine, and that means they are breaking down now. And also you can see the AMP and GMP here, they are converting into guanosine, and AMP not necessarily converting into, no, I think there should be an arrow like this. The AMP converts into adenosine also. And also, as you can see, that adenine with the PRPP, with the help of this enzyme, APRT, adenosine phosphoribosyl transferase, they can convert into AMP. So AMP, that means adenosine monophosphate, can be found through IMP, can be found through this procedure. Now what happens next? This adenosine can convert into inosine by one enzyme that is called ADA, adenosine deaminase. And also you can see this inosine, they can convert or break down more precisely into hypoxanthine. And here you see the guanosine, they can break down into guanine. And you can see, just to take the breakdown procedure, this adenosine converts into inosine, inosine converts into hypoxanthine, guanosine converts into guanine, and all of them can convert into xanthine. This hypoxanthine and guanine, they can convert into xanthine by this enzyme. That is xanthine oxidase. XO is xanthine oxidase. Now this xanthine, with the help of the same enzyme, they can convert into uric acid, and this uric acid can be excreted through renal excretion, or they can convert into far more excretable material called allantoin by the enzyme uricase or PEG uricase. And we have some synthetic drugs that we can use to convert this uric acid into allantoin, and one of the drug is the rasburicase. Okay. Also, you see some other drugs working here that help in excretion of uric acid through the renal system, like probenecid, the benzbromeron, the losartan, the fenofibrates. They can help excrete the uric acid easily through the renal excretion. Now, this is the breakdown procedure. What is the salvage, or what is the recycle procedure? As you can see here, this hypoxanthine and this guanine, with the help of the enzyme HGPRT. What is HGPRT? Hypoxanthine guanine phosphoribosyl transferase. You don't need to remember the full name. If you remember just HGPRT, that's good. Hypoxanthine guanine phosphoribosyltransferase. With the help of this molecule, PRPP, they are converting into respective GMP and IMP. So now, as you can see, that they are recycling. This recycling can give you again the GMP and IMP. That means again the purine can be generated from those breakdown products. And that's it. This is the purine salvage. So what do you need to know from the purine salvage? You need to know some disorders. One of the things is ADA deficiency. As I told you earlier, that here the adenosine can convert into inosine by this enzyme called ADA. And this ADA is adenosine deaminase. And if there is deficiency of this adenosine ADA, suppose ADA is deficient, what will happen? There will be a lot more adenosine. And if it is from the deoxy sugar, there can be deoxyadenosine also. And there can be a lot of deoxyadenosine phosphate. That means deoxyadenosine monophosphate, deoxyadenosine deoxyadenosine triphosphate. And one thing that this DATP, this deoxyadenosine triphosphate, is toxic to lymphocytes. So they are toxic to lymphocyte, and you can understand that when they are toxic to lymphocyte, they can cause damage to the lymphocytes, and which are necessary for our immune system. So one of the immune deficiency disease, that is severe combined immunodeficiency, can be caused by this thing, DATP, deoxyadenosine triphosphate, right? So ADA deficiency can result in this immunodeficiency. Now here you can see the Leschnyhan syndrome, very important and very commonly tested. That what happens here? Excessive uric acid production due to deficiency of AGPRT. Let's go back to the page, and as you can see here, the AGPRT is here. If the AGPRT is deficient, then this guanine and this hypoxanthine cannot really go back and form the purine again. That means the recycling is not happening. You understand the importance of recycle, because if you just keep on accumulating your trash and don't recycle them, what will happen? There'll be trash everywhere. There will be trash everywhere, right? But if you recycle them, you can utilize those trashes. But what happens if you cannot recycle them? There will be a lot of trash, that is uric acid. And remember, uric acid is the cause of gout. We don't want the uric acid, right? So this uric acid accumulates, and this uric acid causes the symptoms of Leschnyhan syndrome. So here in the findings of Leschnyhan syndrome, what do you get? The intellectual disability, uric acid goes into places cause intellectual disability. Self-mutilation, it goes under the skin, and the person is so much agitated. As you can see, aggression, and also gout, dystonia, hyperuricemia. The uric acid comes out through the urine. And if in the baby is so much unfortunate, they got this orange sand crystal in the diaper. And self-mutilation, very aggressive self-mutilation, because they have pain under the skin, and they bite their skin, they bite their hands because they have pain. There is uric acid all over the body. So much, you can say, very unfortunate condition, this Leschnyhan syndrome. Treatment is allopurinol or febugestate. This drug helps in get rid of the uric acid, and also they stop the production of uric acid. As you can see, that allopurinol and febugestate, they stop the production of uric acid. So there's less uric acid in the body. There's some technical question they might sometimes ask you. One of the question is this, that which enzyme activity increases in Leschnyhan syndrome? So this is very intriguing, that whenever you see, suppose the questions are like this, they describe you a vignette. A boy comes to you with this 3-year-old boy comes to you, and the parents notice that this boy is so much pissed off, and this boy has biting behavior of himself, and you do the lab and you found that there is increased uric acid in the body, and you diagnose as Leschnyhan syndrome. It may be given in your vignette, or maybe just the vignette is given and you need to proceed with the diagnosis in the question itself. And after you diagnose, they might ask you which enzyme activity increases in Leschnyhan syndrome. You might think, okay, this is Leschnyhan syndrome, and whenever you see the word enzyme, you go and pick AG purity. But be careful. AG purity activity decreases in Leschnyhan syndrome, or it is deficient in Leschnyhan syndrome. So you need to think that what may be increased in Leschnyhan syndrome, which enzyme activity. It is understandable, just to give you a clue, that this enzyme AGPRD activity is decreased, right? So what will happen? That the salvage pathway is decreased. Salvage pathway is decreased. So the PRPP syndrome, yes, Dr. Thank you, Dr. Tamanna, that she said that this enzyme activity, that means the de novo synthesis pathway, will increase its activity. If you stop the recycling, what do you need? You need more new material. And that's why when you stop this thing, the new pathway or the new de novo synthesis pathway enzyme will increase their activity. And sometimes, as I told you earlier, that these steps are not shown clearly in your book, but sometimes they are necessary. Like as you can see here, sorry, where is the thing? Okay, here you can see that sometimes they might show, all right, this enzyme, they can give you this enzyme named PRPP aminotransferase. This is also a de novo pathway. So they might give you the vignette, they might give you the option, and in the option, they might give you this enzyme name, not PRPP synthetase. They might give you this enzyme named PRPP aminotransferase. So this enzyme activity can increase in this PRPP, in AG purity deficiency. All right, so now let's go to the next page. So this enzyme, some enzyme names is necessary to remember. But main is the concept. If you know that if some procedure is stopped in the meat pathway, the previous enzyme activity will increase, all the materials or all the contents will increase in amount because they cannot proceed further, so they would accumulate. Another thing is tumor lysis syndrome, and in tumor lysis syndrome, what happens that when someone comes with a tumor, suppose you do the induction. What is induction? Induction is when someone comes with a cancer or tumor, you give them a high dose of chemotherapy, and that chemotherapy kills most of the tumor burden. Usually the induction is done like this, that they kill the tumor cell very rapidly and in very large number, like less than 5% of tumor cell will be left after the induction. And then you do the adjuvant therapy, you do the maintenance therapy. These are coming later, but this is induction. So when you do the tumor induction chemotherapy, what will happen that this tumor lysis syndrome may occur? Because what is a tumor? It's a new growth, right? It's a neoplasia, it's a new growth in your body. And when you destroy those tumor cells also got this material. They got the DNA, they got the RNA. And when you break those things, what will happen? Those broken down DNA and RNA will produce purines and pyrimidines, right? And those purines and pyrimidines, when they eventually break down, they will give rise to ultimately uric acid, as I showed earlier, that purines breaking down and eventually converting into uric acid. So uric acid is coming here also, and that can cause symptoms. So here you can see that lysis of tumor cells rapidly due to chemotherapy leads to hyperphosphatemia. Phosphate is rich in DNA. So when you break down some cell, destroy some cell, that cell's DNA breaks down, it releases the phosphate. Those phosphate binds with the calcium already in your body, and the calcium level goes down, and that is hypocalcemia. And cells are rich in potassium, so you got hyperkalemia. And also, as I told you earlier, the purine breakdown products give you hyperuricemia. So ultimately, you need to get rid of the uric acid because that is the most problematic. Also, you need to take care of the other thing like hyperphosphatemia, hyperkalemia. But in this case, in this discussion, allopurinol and rasburicase, they help in get rid of those uric acid. Now, before going to the DNA replication, let's take a break for 5 minutes. This is 5:58 a.m. So we'll start at 6. Let's say it's 6 a.m. We'll start at 6:05. 6 and 5. 6 a.m. and 5 minutes. Okay, let's take a break for 5 minutes. Sorry for that. Now I think you can hear, right? Let me know, let me know. You can hear everyone? Good? Yeah, okay. Sorry for that. I sometimes press this button and it stops the microphone. Sorry for that. All right, so DNA replication is the central dogma of molecular biology or molecular biochemistry. Uh, what is that? That DNA converts into another DNA, or simply DNA makes a copy. DNA makes not only one copy from one DNA, we got two DNAs, and that is the replication or forming of a replica. You probably heard of the term replica, that you went to visit France or Paris, you bring a replica of Eiffel Tower. You go to Manhattan, you buy a replica of the Flatiron District, Flatiron Tower, or Flatiron Building. Okay, Flatiron Building, sorry. Now, here you see in this DNA replication, what is happening. Suppose this is a zipped up DNA, or this is a DNA double helix. And as you can see, they gave some numbers, like here is a 5 prime, and at the end of it is 3 prime. And here is another 5 prime, and at the end of it is 3 prime. This prime and these numbers are the direction of the DNA as their carbons are arranged. One DNA is like this, another DNA is like that. So they are anti-parallel, or one strand is like this, another strand is like that, anti-parallel strand. We need to know this some of the directions, because sometimes they want us to confuse with these directions. So how this direction works and how this replication works. One thing you should remember, that replication, the new strand formation, new synthesis or new strand synthesis, always works in 5 prime to 3 prime direction. So the replication, whatever it is, the leading strand or the lagging strand, the direction is from 5 prime to 3 prime direction. The new strand formation happens like this. So you don't need to remember the other part, because if you want to create a new strand, like as you can see here, 5 prime is here, and this is the new strand that is forming in this way, 5 prime to 3 prime direction. It should have a template strand. Template means the strand on which the new strand will work on and synthesize a new strand, and it will follow the direction the opposite. So you don't need to remember the template strand direction, just remember that replication happens in 5 prime to 3 prime direction. Now, as you can see, the first thing that happened before anything else, or bay, we use the word bay in this modern social communication very much. This before anyone else or before anything else, there is the origin of replication is identified and bound by a multi-subunit protein. So the first task is to identify the identify the origin of replication. That means if you have a DNA a strand, 2 DNA strand, where this replication will begin, that is first identified, and that is done by a very complex protein molecules. We don't need to know, but you can know the name is origin recognition complex. They goes there, they sits there, and they say, okay, this is the place where the DNA replication will begin. And what that does, this proteins, they slightly dissociates these 2 strands. If the 2 strands are here, this slightly dissociates these 2 strands on that specific point. So that, because if these strands are coiled like this, nothing can sit here. So this origin recognition complex goes there, it slightly opens up or slightly dissociates those double-stranded DNA. And then everything else starts. So before anything else, the origin recognition complex recognizes or identifies the origin of the DNA replication. Now this helicase comes into action, and this is the helicase, is the unzipping enzyme. And as you can see, helicase is the enzyme, these are the helicase, and they are unzipping the DNA. They unzip the DNA. And what happens when you unzip the DNA, there's this space. There is this space. Suppose the DNA is like this, and you unzip them. like this. Okay, and what will happen, this area, this Y-shaped area, is called the replication fork. This area is replication fork, and this not only happens in one part. If this is the double strand, in one way you can see two replication fork here, one on the top here and one on the bottom here. Two replication forks are formed here, right? Next, what happens that when the helicase unwinds and forms the replication fork, and there is this single strands, those are formed, right? Suddenly there is some single strands, those are formed, and those single strand has a tendency to go back and cluster together. To prevent that clustering, there is something called single-stranded binding protein. And those single-stranded binding protein binds with those single strand and stabilizes those single strands so that they don't go and don't snap and cluster together, right? Then what happens that there is something called DNA polymerase that comes into action and starts creating the DNA. But before that, you need to have a RNA primer. You need to have a primer. That means a starting point. A starting point is required through which the or depending on which the DNA should work. DNA should work. All right. So what is that thing, the RNA primer thing? It is not shown in this strand on the top strand, but it is shown in the bottom strand. But usually you need the RNA primer. This is the primer, or this is the initiation thing. So suppose you start to create a new DNA, you need that primer thing. That means you need this starting thing. I'm not good at makeup, but my wife, she says that bring some primer for me or something like that. You need to put on some primer on your face before you put those makeups on. So this is kind of like this, that you put the primer, and on top of that you start working to synthesize your new DNA. And this primer is synthesized by something called primase. This primase forms this primer, and on depending on the primer, the DNA polymerase, the enzyme, the DNA polymerase works to synthesize new DNA. And one thing is confusing, that sometimes you see there is a leading strand and there is a lagging strand. So the strand, suppose, as you can see in the top strand, this thing simply, this is following the helicase. If the helicase is going this way, this is unzipping the DNA in this way, right? And this thing, this DNA polymerase, is creating a new strand this way. And this strand is called the leading strand, because this thing is following the unzipping way, or the way of helicase. But this strand that is not following the down strand, as you can see, they are going the opposite way the helicase is going. But they are following a helicase on the other side. There is another helicase here that is working this way. For this aspect, for this relativity, this is the leading strand because this is following this helicase. And this strand, this down strand, is lagging strand because this is going away from the helicase. All right. So helicase are going and unzipping. The DNA polymerase that is synthesizing following the helicase way, that strand is leading strand. The DNA strand that is away from the helicase, that is lagging strand. All right. So which DNA polymerase works here for the synthesis of the new DNA? This is DNA polymerase 3 in case of prokaryotes. Whatever enzyme we discuss here, usually those are for the prokaryotes, not us. We're eukaryotes. The prokaryotes enzyme. The eukaryotes enzyme in this case are much more complicated. They are divided into alpha, beta, gamma, and this kind of subtypes. But we usually discuss the prokaryotes. What I said, DNA polymerase 3, that synthesizes these new strands. Now, one thing you should remember, that this lagging strand, suppose here you can see a lagging strand, and lagging means what? In relative to this helicase, these strands are going away. They are not following the helicase. So that's why they are called lagging strand. They are lagged behind or something like that. And as you can see, there's these lagging strand, they are in fragments. They are not continuous. They are from like this, another lagging strand like this, another lagging strand like this. These fragments are called the Okazaki fragment. Okazaki fragments, and what happens, these Okazaki fragments, they have these holes. These holes are then sealed. After everything is done, they are sealed by something called ligase. This is some basic discussion of the DNA replication, how this thing happens. Now, what do you need to know? You need to know some enzymes. One of the enzymes is DNA topoisomerase, and they comes in different flavor, type 1, type 2, type 3, type 4, different flavor of topoisomerase, but usually their basic of action or basic mechanism is same. So what is this thing, this topoisomerase? So when you go back and look at this DNA getting unwind, you can understand, if I can show you with some wires. Suppose this is a DNA that is coiled here, right? This is a DNA that is coiled here. And when you try to unwind it, here, as you can see, there is a tightness. There is a tightness here. This is unwinding, but there is a tightness is approaching here or occurring here in this part of the DNA. This is called the supercoiling. This tightness, as you can see, this is called the supercoiling. When you try to unzip a DNA, there is a supercoiling that is happening here. And you need to get rid of that supercoiling. And how do you get rid of that? You get rid of that by this enzyme called DNA topoisomerase. As you can see that there is some coiling here in this DNA. There is some coiling, and this enzyme, this topoisomerase, what it does, they just cut this one strand. The type 1 cuts one strand and type 2 cuts two strands of the DNA. So what happens when there is some supercoiling? You cut a strand, that pressure is released. And when the pressure is released, again, this same enzyme ligate it. So this is a cutting and joining enzyme. So first it cuts the one strand or two strand, and then it again reseal them. This is the way this type 1, or you can say that type 2 topoisomerase, DNA topoisomerase works. Now here's something written about this topoisomerase and some drugs we can use in our favor to prevent this topoisomerase from working and to prevent some cell from replicating. Because the thing is, this learning of replication, why you need to learn them? One, you need to pass your ASML step 1 exam, because they will ask you questions from this replication. Another is, if you know this thing, okay, DNA is replicated like this, and you know how the pharmacology or pharmacotherapy works. Like here, you know that these drugs in eukaryotes, irinotecan, topotecan, or etoposide, tiniposide, these are anti-cancer drugs. They can inhibit this topoisomerase. And what will happen if you inhibit those topoisomerase? Those DNA, suppose you have a tumor cell, that tumor cell is replicating continuously. And during replication, there is some supercoiling formation, right? And that topoisomerase is getting rid of that supercoiling so that that tumor cell can replicate easily. But if you block that topoisomerase, that supercoiling cannot be get rid of, or it cannot be released. So what will happen? That tumor cell cannot replicate. And when it cannot replicate, it will die. So that's the way this pharmacotherapy works. And also in prokaryotes, as you can see, that fluoroquinolones are the drugs that inhibit this topoisomerase 2, also called DNA gyrase, and topoisomerase 4. You don't need to know very minute details of different topoisomerase. Just remember that they cut and they reseal. They cut and they reseal. And in case of prokaryotes, they have this name, DNA gyrase. DNA gyrase or gyrase, whatever you say, the prokaryotes topoisomerase, which can be inhibited by the very famous drug, the fluoroquinolones. Now, one thing is important, the direction of activity of DNA polymerase. Now, go back to the picture that, as you can see, that DNA polymerase is working here, right? This is the DNA polymerase, or this is the DNA polymerase. They are synthesizing a new strand from this direction, 5' to 3', right? They're synthesizing a new strand. Now, this DNA polymerase has different activity. This replication procedure is very sophisticated, very sophisticated. It cannot make a mistake. If it makes a mistake somehow, mistakes happens. If somehow mistakes happens, it has a proofreading function. That's very important. So you need to know where this DNA polymerase is working in a synthesizing direction. You need to know how they are proofreading, in which direction they are proofreading. You need to know both. So here I wrote that DNA polymerase has 5' to 3' synthesis activity, and it can proofread 3' to 5'. So this DNA polymerase, it proceeds from this direction, from 5' to 3', it is proceeding. That's very good. But what it does, it goes this way, 5' to 3', and then it turns back to see whether there was any mistake. What can be the mistake? Suppose here is adenine, and here should be thymine, right? But if somehow by mistake there is some guanine or some cytosine, this DNA polymerase will go, it will come back, it will repair it, then it will again go to its synthesis procedure. This is called DNA proofreading function, and that is done by the polymerases. DNA polymerase 1, DNA polymerase 3, they have this function. So, very important, DNA polymerase 3 has 5' to 3' synthesis function, and 3' to 5' proofreading function. Very important. And whenever you read it in your book, you will remember my word. Now, another polymerase is DNA polymerase 1. This DNA polymerase 1 is important because you remember the RNA primers. As I told you earlier, you need to put some primer before you do the makeup. Like you need to put some RNA primer before you start synthesizing a new DNA. This DNA polymerase 1, what it does, it get rid of these primers, because this is a DNA, right? At the end of the day, this is a DNA. We don't need any RNA here. This is RNA. What does it mean? It means it has uracil, right? Sometimes the questions might ask you that a scientist is researching, and they ask a lot of questions like this. A scientist is researching this, a scientist is researching that, and they found this and that, and they ask a question from that thing. A lot of vignette you'll see that starts with this same thing. A scientist or a medical student is researching this thing. So they might ask you that they are researching a replicating model from a prokaryotes, and they are researching a DNA. They're researching a DNA, and they found some uracil there. What may be the cause? Why there is uracil in the DNA? And if you know this procedure, that in replication, you need a primer, and a RNA primer. So when there is a RNA primer, there is a RNA, and RNA must contain uracil. So if you find a uracil in a DNA which is replicating, that must be a RNA primer. But at the end of the day, you don't need that primer. You need to get rid of that primer. You just used it, and you need to get rid of that primer, right? So how you get rid of that? I think you cannot get rid of the makeup primer though. If you put the primer, you put the makeup on top of that, you brushed yourself, and then you cannot get rid of the primer, right? But in this case, at the end of the day, you need to get rid of the primer. So how you do it? The DNA polymerase 1 is the guy which helps you do it. DNA polymerase 1. So this has a very interesting and very important way of action. This DNA polymerase 1 that degrades the RNA primer in 5' to 3' direction. Very important. 5' to 3' direction, not 3' to 5'. 5' to 3' direction. Go back to the picture so it becomes clear to you that this is the primer, right? This red thing is the primer. A DNA polymerase comes here, and this is the 5', this is the 3', right? 5' to 3'. So this DNA polymerase get rid of the thing in this direction. From 5' to 3' direction, they get rid of the primer. And then what happens? It has the same function as the DNA polymerase 3. So DNA polymerase 1, it removes the primer in 5' to 3' direction, and it has the same function of DNA polymerase 3. So what is it? Let's go back to the DNA polymerase 3 first. DNA polymerase 3 has 5' to 3' synthesis activity, and it can proofread 3' to 5'. So on top of that, the DNA polymerase 1, it has another activity. That is, it degrades RNA primer in 5' to 3' direction. And it can synthesize new DNA in 5' to 3' direction. It can proofread in 3' to 5' direction. I know this is confusing, just to give you the idea again. So when you are synthesizing a new DNA, suppose these are the two strands, and this strand on top of that, you are synthesizing a new DNA. Let's say this is the DNA polymerase 3, all right? So this is the 5' end, and this is the 3' end. This is the 3' end of the template, and this is the 5' end of the template. And this is the DNA polymerase 3, and it will synthesize a new strand in 5' to 3' direction. The new strand, not the template. The new strand will be synthesized in 5' to 3' direction. Now, when it proceeds like this, it goes into 5' to 3' direction, new strand synthesis. It comes back in 3' to 5' direction. It proofreads, and then it again goes back into 5' to 3' direction. Now, DNA polymerase 1, on the other hand, what it does, it gets rid of the primers. So suppose here is a primer here. Here is a primer. So first, it will get rid of it in 5' to 3' direction. First, it gets rid of the primer in 5' to 3' direction, and then it creates a new DNA in 5' to 3' direction. Again, it goes back to proofread in 3' to 5' direction, and again, it proceeds to 5' to 3' direction. So the DNA polymerase 1 has one extra activity of excising or degrading the RNA primer in 5' to 3' direction. It can be confusing, but if you look at the book and read those, it will be easier, much more easier. Just think about it, okay? Because they want to confuse you. Trust me, they want to confuse you in the books and in the, not in the books, in the question, and they want to confuse you on this thing. All right, so the next thing is the telomerase. What is telomerase? Telomerase is the enzyme that is necessary to elongate the tail, of the DNA. So you might ask me, what is the necessity for elongation of the tail? All right, to tell you and to describe you in more detail, that suppose here is a DNA strand, and here is another strand of that DNA, and these strands are getting replicated. Suppose this red strand is getting replicated, and this is the DNA polymerase. Just take into account one polymerase. And what will happen that this DNA polymerase will come all the way here, but it will not replicate the full length. It will not replicate the full length. It might stop 3 or 4 bases or maybe 5 bases earlier. So not all the bases are touched. If the length is take into account in the numbers, if the length is 10 nanometer, this strand, it will do, this DNA polymerase will replicate up to 9 nanometer. So what will happen? The new strand will be a little bit shorter, right? The new strand will be a little bit shorter than the older, the mother strand. And it is happening in our body. This thing is happening in our body every time we are replicating a DNA. Every time our new strand of DNA is getting shorter and shorter and shorter and shorter and shorter, and it vanishes. No, it does not vanish. Why? Why it does not vanish? Because there is something called telomerase or tail elongation thing that elongates that shortened strand. So what is the function of telomerase? This is the new strand that is formed from an old strand, and this is the old strand. And as you can see, the new strand is a little bit shorter than the old strand. Now the telomerase comes here, and by the way, this is an RNA-dependent DNA polymerase. Again, RNA-dependent DNA polymerase. That means it needs a RNA template to work on. It cannot work by itself. It needs an RNA template. This telomerase elongates this shortened strand a little bit, and it almost resizes the new strand with the old strand, almost. That means yet our new strand is shorter, a little bit shorter than the old strand. So the RNA, the telomerase is an elongation enzyme, which increases the size of the new strand a little bit, but it cannot completely elongate the new strand to the size of the old strand. It tries to do it, but it cannot fully do it. And one thing I just said, it's a RNA-dependent DNA polymerase. So where is the RNA? You say, okay, there was some primase enzyme that was creating a new RNA primer. On top of that, the DNA polymerase can work. But where is the RNA in case of telomerase? Telomerase is a complete package. Telomerase comes with everything, right? It's ready. It's always ready. So this telomerase is composed of 2 main structures. One is called the TERT, TERT. Another is the TERT. So what is TERT and TERT? You don't need to know the full form. This TERT is a reverse transcriptase. What is reverse transcriptase? Another new word for you. Transcription, before knowing the reverse transcriptase, you need to know what is transcription. Transcription is forming a RNA from a DNA. So DNA converts into RNA, and this procedure is called transcription. So what is reverse transcription? Simple. To form DNA from an RNA. If you reverse the procedure, this is reverse transcription. Now this enzyme, this telomerase, is a package. It comes with a reverse transcriptase, and as I told you earlier, it's an RNA-dependent enzyme. It comes with its own RNA template. As you can see in this picture, it has a RNA template with it, and it has this nucleotide, this enzyme part with it. So it comes with a package. It comes with a RNA template. It comes with a reverse transcriptase. So you just put the RNA template on the end of that shortened DNA and just elongate a little bit. Just elongate a little bit. Simple. Now, let's come to the mutation. You might ask me, okay, so there is so high fidelity or highly sophisticated procedure through which DNA is replicated, but yet, yes, there can be mistakes. There can be mistakes and there can be mutations. And usually we discuss 2 types of mutation: point mutation and frame shift mutation. There is another type of mutation called splice site mutation. We're going to discuss it in the RNA section, because the RNA, while they're transcribing into DNA, while they're transcribing into RNA, there can be some mutation. Now, this mutation, the point mutation and frame shift mutations, the point mutations can be divided into these subtypes. Like here you can see in this chart, this is the DNA level, this is the mRNA level. That means DNA to mRNA, usually the mistakes happens on that phase. And here's the protein level. So again, to remind you, from DNA to DNA, the procedure is called replication. From DNA to RNA, transcription. RNA to protein, translation. DNA to DNA replication, DNA to RNA transcription, RNA to protein translation. Now here you can see that there is no mutation, and this is the DNA genome, and this is the mRNA that is formed from this transcription, and this is the protein that we get from this mRNA translation. But there can be a silent mutation, there can be a nonsense mutation, and there can be a missense mutation, and those are point mutations. So what is silent mutation? Silent mutation is, as you can see here, this nucleotide is changed. Instead of C, there is T. But here, as the nucleotide is changed, the transcription will also result in change. Instead of G, there is A. But look at the protein, it's the same protein. These proteins, when you go into the protein discussion, you'll see that this genetic code, a lot of genetic code, like there are, if I say one amino acid, suppose lysine, right? This lysine can be found from different genetic codes. This is called genetic ambiguity, that lysine can be found from different genetic codes. But one genetic code can give only one amino acid. That means if this is the genetic code TTT, it can only produce lysine. It cannot produce arginine. TTC, it can only produce lysine. It cannot produce anything else like histidine. It cannot produce that. But lysine, as you can see here, it can be found from TTC, it can be found from TTT. This is a property of the genetic codes. Now, as you can see here, there is a mutation, there is a change, but still the protein is same. So there is silent mutation. The mutation cannot be shown. This is called silent mutation. What is nonsense mutation? Nonsense mutation, look at here. This T is converted into A, and this A is converted into U. In UAG is a stop codon. When you discuss a codon, you see that there's some start codon, there's some stop codon. Whenever there is a start codon, the new protein synthesis begins. Whenever there is a stop codon, there is certain stop codons. One of them is UAG, another one is UGA, and another one is UAA. UAA, UAG, UGA. So these stop codons, whenever they're encountered by the protein synthesizing material, the tRNAs and the ribosomes, they stop synthesizing protein. In this mutation, in nonsense mutation, the mutation is happened in a way that the protein synthesis is stopped. So this is a nonsense mutation. What is missense mutation? Missense mutation can be conservative, can be non-conservative. Conservative means you have a mutation, suppose this T is converted into C and this A is converted into G, but you have an amino acid that is similar in nature to your intended amino acid. You were intending for lysine, right? But you got arginine. It is almost similar in chemical nature. So you can work on this, work with this also. It's kind of sugar and Splenda. You don't have sugar, you can use the Splenda. So this thing is called conservative mutation. That means your amino acid, the new amino acid that is formed, that is not your intended amino acid, but this is same in chemical formula or chemical nature, so it doesn't matter. what amino acid you got. And another missense mutation is the non-conservative mutation. In non-conservative mutation, there is something called a new amino acid is formed that is not intended and that is not similar in chemical nature. So as you can see here, instead of lysine, you get threonine, and you don't need threonine here, or you don't want threonine here. And as you can see that this colors that defines that threonine is a polar amino acid and arginine is a basic amino acid. So they are not matching with their chemical nature also. So this is missense non-conservative mutation. So these are the point mutation that you might get: silent, nonsense, missense. In case of frame shift mutation, what happens? The frame is shifted. If you look at this picture, you can understand that what is frame shifting, that here is the DNA base CAT, 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 C, a lot of CAT, cat. And these are the amino acid: histidine, 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 histidine. All right. If you insert a single amino acid here, instead of this place someone is inserted like this, and adenine is here, and look, all the sequence is changed. So if you insert or if you delete some amino acid from a sequence, if you insert or delete a nucleotide from a sequence, this is called a frame shift mutation. But there's a rule, very important, and that is sometimes confusing, that to be a frame shift mutation, there should be insertion or deletion of number of nucleotides not divisible by 3. Either, suppose here in this example, you are inserting one nucleotide here, right? You cannot insert, if you had inserted 3 nucleotide, that won't be a frame shift mutation. Because if you inserted 3 nucleotide, there will be a new amino acid, but the other amino acids will not shift their frame. That sometimes it happens that sometimes 3 nucleotide are inserted. What is that called? That is called in-frame mutation. So when you have a frame shift mutation, it should be some number of insertion or deletion that cannot be divisible by 3. If I say, let's take into example, if 8 amino acids, 8 nucleotides are inserted here, 8 nucleotides, what will happen? It will be a frame shift mutation. If I say 8 nucleotides are deleted from here, it will be a frame shift mutation. But if I say 9 nucleotides is inserted here, what will happen? This will be in frame mutation because 9 is divisible by 3. So remember, in case you, in order to call it a frame shift mutation, the insertion or deletion number should not be divisible by 3. It can be 8, it can be 11, not 9, not 12, not 15, not 18, or so on. Okay, be careful. Now, this is called something called the lac operon. The lac operon, also called the lactose operator genome. What happens here? Our friend E. coli. We love E. coli, right? We have E. coli, lots of E. coli. This E. coli prefers glucose as a food. The E. coli E. coli prefers glucose as food, but when glucose is not available, suppose in the environment the glucose is not there, what it does, it shifts its genetic machinery in a way that it can utilize lactose. So instead of glucose, it's now using lactose. This genetic machinery shifting is done by the lac operon. It's a sequence of gene that helps in conversion from a glucose utilizing E. coli to a lactose utilizing E. coli. So what is this? What is this lag operon? As you can see here, that this is very important, sometimes tested on your questions, that here is the lag genes, lag Z, lag Y, and lag A. But this lag genes needs to have an operator. This operator is something like triggering. You need an operator to run anything. You need an operator to run a computer. You need to run an operator to run a Tesla car. But sometimes you say, okay, operator, Tesla does not need a driver. But yes, there's some robotics that is operating the Tesla. You need operator everywhere. So this operator is sitting here, and when it is triggered or when it is free, it can run or it can transcribe this gene, this lag Z, lag Y, and lag A. You might ask me, what is this gene? These genes are necessary for producing enzymes. Those are necessary for lactose utilization. So E. coli was dependent on glucose, that's good. But when the glucose is not there, but lactose is there, they need to have the enzyme to utilize the lactose, right? And those enzymes are formed by these genes. Now here also you can see that there's a promoter region. This promoter region is the region where the transcription occurs, or transcription initiates, you can say. What is transcription? Again, from DNA to RNA formation is called transcription. And we're going to discuss it. RNA polymerase are the enzymes that binds with the promoter and initiates the transcription. Now, as you can see here, there's a cap site, catabolite activator protein binding site. And there is some protein called catabolite activator protein that binds in the cap site and promotes RNA polymerase binding. So what is this cap thing? It's a protein that is found in the environment of the E. coli inner environment. And what happens that when there is less glucose in the body, this cap protein activates. This cap protein activates, and that binds to the cap site. That cap site stimulates the binding of polymerase to bind here in the promoter, and then this operator takes into account and goes on creating new enzymes. But one thing is important. Here you see some repressor. The E. coli has some repressor proteins that stops the operator from working. So when everything is there, suppose glucose is not there, so the cap is there. Cap can bind here, and cap can promote the RNA polymerase to bind to the promoter site. One extra thing is necessary, and that is to remove the repressor. And our lactose, if there is lactose in the environment, that lactose can remove the repressor. Some lactose are used to remove the repressor so that the operator can work in a flow. So in a nutshell, what is this lag operon? It's a genetic sequence through which the E. coli changes its machinery from glucose utilizing to the lactose utilizing. So when there is less glucose in the environment, there is some change. Suppose there is less glucose in the environment, there is cap which can bind to the cap site. There is RNA polymerase which can bind to the promoter, and there is some operator which is bound to the repressor. But when there is lactose, this lactose can remove this repressor, like as you can see in this second picture, the yellow lactose is removing the repressor, and the operator can work on transcribing this gene, which are necessary for lactose utilization. And that's the way E. coli shifts from glucose utilizing moiety to lactose utilizing moiety. So this is very, very nice. Now here you can see that what is the control of the lag operon and its control element. Like if you have this thing, if you have this requirements, then the lactose will work. If you don't have the lactose and this glucose, it will not work. Let's see what is this thing. So here is the lag operon thing. Here is the cap binding site. Here's the promoter. Here's the operator. And here's the genes, right? Now, when there is low glucose and lactose available, when there's low glucose, what will happen? This cap protein can bind here. One thing you should remember that when you go and read in the book, that if there is glucose, this cap protein cannot be activated. If there is glucose, this cap protein cannot be activated. But when there is low glucose, this cap protein can activate. This RNA polymerase can work. And also the repressors cannot bind here. So the lag genes are strongly expressed. If there is high glucose and lactose is unavailable, then what is the need for this lag operon thing? So lag genes are not expressed. You don't need those genes now because you have the glucose now, right? When there is low glucose and lactose is not available, what will happen? The cap can bind here, but there is the repressor. And remember, you need the lactose to remove the repressor. So although you have the cap, although you have low glucose, but as there is no lactose, the lag genes is not expressed because you cannot remove this repressor gene. When there is high glucose, when there is also lactose available, everything is available, the E. coli is now in peace of mind. It can have, oh, well, I have glucose in efficient amount and sufficient amount, and I have also lactose, so let's express some of this. And there's very basal level of gene expression, not really that much, but it can express the gene. So this is the control element of lag operon expression. Now, very important, that DNA repair. We discussed that when the DNA synthesis was happening, the replication was happening, there was very sophisticated procedure and there was no mistake because the DNA polymerase has proofreading activity and this and that. But still, there can be mistakes. And those mistakes are repaired by some repairing mechanism. If there is a single strand problem, there is some repairing mechanism. These three mechanisms are important: nucleotide excision repair, base excision repair, mismatch repair. We're going to discuss it now. First one is the nucleotide excision repair. Suppose you have a newly synthesized DNA, but after the DNA is synthesized, somehow, as you can see, UV radiation. There is always UV radiation in our skin, right? Those UV radiation can cause some conformational change in your DNA structure. As you can see here, in this DNA structure, a UV radiation fall, and these two thymine, they are together, they're side by side. But they does not have a connection in between. But if there is some radiation, what will happen, that this thymine may cause some dimer formation. That means they will create some bond in between them. You don't need a bond in between the thymine because the thymine will bind with the adenine, that's good, but you don't need them to have bond inside or in between them. If this thing happens, you need to correct it because this will cause problem, this thymine dimer. And if you don't have the correction material, there will be some disease. We're going to discuss it. So how it is corrected? First, once the dimer has been detected, there is detection material or detection procedure. And this is very sophisticated and very amazing, that when there is this problem, suppose in one strand, there is these enzymes, these correction enzymes, these DNA nucleotide excision enzymes, they scrutinize the whole thing, whole new sequence, that to see if there is any problem there. And when they find the problem, they just correct it and repair it. But you might ask me, well, there are two strands there, right? How they know which strand is the new one and which strand is the old one? Is there anyone can answer this question, that how this repair mechanism or repair enzyme can know which one is the old one and which one is the new one? Who can answer? Anyone? My question is, there is a new strand and there was an old strand. The new strand is formed by the replication. Now, repair mechanism comes and it detects the new strand and it works on the new strand. So how it recognizes the new strand? There is something that happened in the old strand where through which they recognize, okay, this is the old strand and this is the new strand. And that is very important. The way they recognize the old strand mutation, no, not really mutation or mismatch. The thing is, let me describe how they recognize the old strand. Suppose this is the old strand, right? This one is the old strand and this one is the new strand. When this repair mechanism comes, they find they correctly detect this is the new strand and they scrutinize the new strand. How they do it? Because in the old strand, there is adenine and cytosine. Let me see. Adenine and cytosine, some of the adenines and some of the, suppose this is cytosine, some of the adenine and some of the cytosines are methylated. In the old strand, some of the adenines and some of the cytosines are methylated. They are like tags that these are old thing. These are old strands. So when the new DNA correcting material comes and they see, okay, these are the old strand, these adenine and cytosines are methylated, so this should be the old strand. They now shift to the other strand and they say, okay, this is the new strand. So what I said, that some of the adenine and some of the cytosines are methylated so that when the repair mechanism comes, they see, okay, this is the old strand and this is the new strand, and they find, is there any problem there in the new strand? Now how they proceed? They detect the problem, and what they does, they create a space like this surrounding the problem. This is the problem and they surround the problem, right? And there is some enzyme. There is some enzyme that comes here. What is the enzyme name? Endonuclease. Endonuclease enzyme comes and they cut these strands here. On the side of the problem, they cut these strands. And they remove this strand, this problematic strand, and a polymerase enzyme comes. They form a new strand here, and they seal these strands with the ligase enzyme. So how it happens? There is a problem in nucleotide excision repair. This is the nucleotide excision repair system. A excision enzyme comes, the endonuclease comes. It detects that there is a problem. It cuts on the side, on the periphery of the problem. It removes that thing. It creates a new strand here. It seals it. So you need to remember the sequence. Which enzyme comes when, which enzyme comes later. Sometimes the question asks the sequence. One of the important diseases is xeroderma pigmentosum. Because this thing, this thymine dimer formation is happening with you, with me, with everyone. But we have the repair system. But if someone, unfortunate, don't have the repair system, they have this problem, xeroderma pigmentosum, and this is the autosomal recessive disease. This thing, this xeroderma pigmentosum, is 8 types. We don't need to know everything. There is photosensitivity, poikiloderma, hyperpigmentation, increased risk of skin cancer. These are the findings of xeroderma pigmentosum, where there is defect or absence of UV-specific endonuclease, or simply there is defect in this DNA repair system. Next is the base excision repair. What happens here? If there is some, suppose we have some base, right? So here you can see that deamination converts the cytosine base into uracil. We know that these bases can be converted from each other, converted to each other by some chemical change. If somehow a chemical reaction occurred in the DNA, and you see there was a cytosine here, but somehow it is converted into a uracil now, because there was some deamination happened due to any chemical reaction, chemical irritation, or whatever. You need to get rid of that base, right? And this is called base excision repair. How it happens? First, there will be a base-specific endonuclease. that comes here. And what it does, they remove the base first. They will remove the whole base. And then they will remove this remaining sequence, because the base, we know that here in this particular part, there is a base, there is a sugar, there is phosphate. First, the base will be removed. That means the nitrogenous base will be removed, whether it is a purine or pyrimidine. Then this whole thing with the endonuclease will be removed. Then a polymerase will come and create a new strand, and again, seal this strand with the ligase. So again, you need to remember the sequence. The base is removed, the nucleotide is removed, then there is there is new strand formation, and there is resealing of the periphery. So that is base excision, remember, base excision repair. So here is the sequence. First, there is glycosylase. Then there is APN. This glycosylase removes the base that is changed or deaminated. Then AP endonuclease. AP means apurinic or apyrimidinic, because when you remove the base, there is no purine or no pyrimidine. You remove that part. Then lyase, that is very important, because on the 3' end, sorry, okay, I think, okay, AP endonuclease cleaves on the 5' end. That is important. And the lyase cleaves on the 3' end. Sometimes they test you on this thing. First, the glycosylase removes the base. Then on the 5' end, the AP endonuclease cleaves. On the 3' end, the lyase cleaves. This thing is removed. A DNA polymerase comes and create a new sequence, and DNA ligase seals the gap. So remember this sequence. Next thing, the last repair system is the mismatch repair. What happens? This is much more simpler. As you can see, if sometimes any mismatch happens, as you can see, here's a G and it binds with a T, but it should be G binds with C, right? But G here binds with T. So it needs to be removed. And this area surrounding the area is cut with some endonuclease and with some mismatch repair enzyme, you can say it. Then there is this replacement with some DNA polymerase. Then there is resealing of the gap with the ligase. Now, here several gene works in this step, and the mismatch repair system involves several genes, including MSH2 and MLH1. Sometimes they test you, so you need to memorize these names, MSH2 and MLH1. And this system codes for a component of the human mut-S and mut-L homologs. These are the proteins that work as a mismatch repair system. If there is mutation in these genes, they will cause something called Lynch syndrome. What is Lynch syndrome? There is one condition which is called hereditary polyposis colorectal cancer. But here, there is a colorectal cancer, and that is hereditary non-polyposis colorectal cancer. That means this is also hereditary. Very good, thank you. This is also hereditary, but this is non-polypotic. That means there is not so much polyps in this condition. One is the hereditary polyposis colorectal cancer, and this is hereditary non-polyposis colorectal cancer. And this is called Lynch syndrome. And that happens if there is mutation in these genes. So these were the single strand repair, and here is one double strand repair. This is much more simpler. If there is break in the double strands, either you join the two strands randomly, or you join the strands with a homolog. That means, what is homolog? That means if the two strands are just joined together randomly, you don't need a complementary strand. Like you don't need A and the opposite side, you don't need a T. You just match them. It's kind of temporary healing. This is called non-homologous end joining. Non-homologous end joining. And if you join it very minutely, like you thing this repair system sees that, okay, this is complementary strand, this is strand, and they correctly and minutely joins them. This is homologous recombination. So double strand repair can happen in homologous recombination, in non-homologous end joining, and some disease are associated with them. Now, these are DNA, all about the DNAs, and now here we go into the transcription and translation. Again, transcription is formation of RNA from DNA, and translation is making of protein from that RNA. I wrote DNA, but ultimately that is right. From DNA, you make RNA, from RNA, you make protein. And that procedure from RNA to protein is called translation. So you can correct it here that translation is making protein from RNA. This should be RNA. All right, let me correct it here so that you get in your copy. All right, so now here you see that a functional sequence of DNA to RNA to protein, just a overview. And here's the DNA, and it has many reasons like enhancer, like control reason, promoter. Some of them are called exons, some of them are called intron, exon, and terminator. When there is transcription, there is first a primary transcript. This is called a pre-mRNA. That means this is a nascent RNA from where you'll get a good or more precise, more mature RNA, right? So now this RNA processing happens. There is removal of this some sequence. Introns are removed actually, and exons are remaining, and they form the mRNA. All right, so we're gonna discuss it in more detail when we discuss the transcription procedure. Now here in this picture, you'll see that some enhancer and some enhancer material in the DNA. And as you can see, this is the enhancer and this is the promoter from where the transcription will begin. So from promoter, the transcription begins. But this enhancer, as you can see, this can bend like this. This can bend like this, and this enhancer can sit here and enhance this transcription. So this enhancer, it can be anywhere in the DNA. It can be inside the DNA, which is getting transcribed. It can be outside DNA like this picture, or it can be within an intron, which will never be transcribed. Usually the introns are transcribed, but they are never be used. So these enhancer materials, or there is another thing that is called silencer. These are materials, they can be outside the DNA, they can be inside the DNA itself, and they can be inside the introns also. So if they ask you a question because they try to confuse you, that there is a sequence of codes that is situated 300 base pair away from the promoter region, which can be this. It can be an enhancer, it can be a silencer. So don't get confused. If they are outside of the DNA, still they can bend and they can cause this change. And as you can see, there's a protein that is helping them, DNA bending protein, that bends them and utilizes them where they are needed. Here's the RNA polymerase types for eukaryotes, because in prokaryotes, there is only one RNA polymerase, which synthesizes all of the RNAs. In the eukaryotes, there's 3 forms of RNA polymerase. RNA polymerase 1, 2, 3. There is several other types, but for step 1 purpose, just remember this 3, 1, 2, 3. And what do you need to remember? Where they, what are their products, where they work. Very important. The rRNA, or the ribosomal RNA, is synthesized by RNA polymerase 1, and they work on the nucleolus. Remember it, nucleolus, not nucleus, nucleolus. RNA polymerase 2, they synthesize the most abundant mRNA and also some portion of snRNA, the small nuclear RNA, which is needed for the RNA post-transcriptional modification. This mRNA and snRNA, these are formed in nucleoplasm. And RNA polymerase 3, they create a portion of rRNA, ribosomal RNA, 5-base RNA, and tRNA. And that is also located in nucleoplasm. But if you forget it, because I always forget this thing, what do you need to remember? Just 1, 2, 3, and the very important thing, like 1 R, 2 M, 3 T. And if you look at them, that M and T, they follows the alphabetic order, but 1, they not follow the alphabetic order. The R comes first, then M, then T, RMT. 1, 2, 3, RMT. 1, 2, 3, RMT, something like that. Okay, the next thing is some words about amatoxin or alpha amanitin. This is found in death cap mushrooms or amanita phalloides. What it does, if someone eats this mushroom, somehow someone is walking down the road and sees some mushroom and see, okay, that's a mushroom, I want to eat this, and picks it up and eat it. Sometimes it happens that this is a death cap mushroom and it contains this toxin called the amatoxin or alpha amanitin. What it does, it inhibits the RNA polymerase 2. How it occurs? Well, when it is absorbed in the GIT, this should be GIT. I had so many mistakes, sorry for that. After absorption by the GIT, this amatoxin are transported to the liver via portal circulation, where active transport by OATP and NTCP. Those are transport and names. You don't need to remember the names. OATP and NTCP, although I wrote the names, you don't need to remember the names. From the liver, from the portal circulation, they are transported to the liver, and they, you know that our liver cell works with everything, the toxins and everything. But what it does, it toxifies the liver cell, and it causes the blockage of the RNA polymerase 2. Very important thing, the mRNA cannot be processed, so there will be symptoms, of course. And the symptoms typically starts within 6 to 24 hours. Abdominal pain, vomiting, severe cholera-like diarrhea that may contain blood and mucus and can lead to hepatic and renal failure. So what kind of question can be asked from here? There can be a history of death cap mushroom ingestion, and there is this symptoms, abdominal pain, vomiting, and this, and they might ask you how this toxin works, and you know that they blocks the RNA polymerase 2. Okay, so that's it for today. We reached our saturation, and do you have any question up to this? This video will be uploaded in the YouTube. This is the first video that will be uploaded in the YouTube. But from tomorrow, the videos will be uploaded only in the cloud. That means only in the Zoom. So those who are registered can see this. But this video will be uploaded in the YouTube with a link, with a shareable link, although private, but that can be shareable. So do you have any question up to this? Any question? Let's see. Okay, my pen is not working. Let's see. Chat. Okay, no thanks. Great effort. Okay, thank you very much. I was trying to go a little faster because, as I told, my grandma is yes, I will send the link. No worries. I will send the link to everyone. Pray for my grandma because she is a strong lady, but I don't know what happened. My aunt called and say that she is having vomiting, severe vomiting. So I need to go there now. Fortunately, I'm awake now because I usually sleep. So this is kind of a fortune. All right, so I will send the video. I'll send the video link, and yes, thank you very much for wishing well for my grandma. And let's stop for now. We will meet tomorrow, same time, 5 a.m. and we'll continue our discussion. And I'll send you the videos, don't worry. Take care, everyone. Goodbye.